Hello, everybody. Thank you for giving me the opportunity to present our work and contribution to the reproducibility concept in this conference. My name is Haseli, and I'm a senior data scientist / statistician at European Bioinformatics Institute, EBI. In this talk, I'll go through the reproducibility in the statistical analysis of high-throughput phenotypic data with a focus on the International Mouse Phenotyping Consortium. Okay. All right, let's start. What is IMPC? IMPC stands for International Mouse Phenotyping Consortium. We in the IMPC aiming to identify the function of every protein coding gene in the mouse genome. Precisely, IMPC is an international effort by 21 research institutes globally, as you see in this chart. We're now celebrating the 10 years anniversary of the IMPC. We in the IMPC produce several types of data, such as categorical data, images, text, time series, etc. In the current release of data release 15.1, we produce about 61 million plus data points. And everything that we produce is publicly available from the IMPC web portal or the endpoints. All right, what are measured and how we stratify the type of measurements? First, we should know that there are two types of inputs to the IMPC mouse production pipeline: wild types and knockouts or mutants. We use the animal with the same background strain and pretty much clone of each other for controls for this group. For the mutants, the process is to knock out a gene from an allele to create mutants. The whole pipeline can be broken up into 3 stages: embryonic stage, in life stage, and terminal stage. As you see from the chart, the measurements are performed based on the age of the animals in weeks, like here, like all of these numbers that are age in weeks, or the last terminal week, which is 16 weeks. In the end of 16 weeks, the animals will be terminated and will be tested for internal organs such as heart rate. As we talk about the procedures and parameters in the IMPC, like ECG and these kind of things, let's take a look at one of these, which is here, ECG for electrocardiogram. A very detailed description of the process is kept in one of our infrastructure called IMPRESS. IMPRESS stands for International Mouse Phenotyping Resource and Standardized Screens. As you see on the left-hand side, in IMPRESS, there is a good explanation of the experiment, the inputs here, 5 males and 5 females at the age of 12 weeks. We expect the sexual dimorphism in our data, etc. Plus, we have a good description of the procedure, plus some charts, and how and very detailed descriptions like anesthesia procedures and plots, charts. Here you see the RR chart, PQ, RST charts, and based on these experiments, we have some measurements. All of these measurements are assigned an ID. As you see on the right-hand side, like here, for example, waveform image has IMPC ECG 025001 ID. We also collect a very detailed metadata of the experiment. Here you see the equipment ID, or very fine details such as analysis software, last calibration dates, even the configurations, etc. In IMPC, overall, all of these experiments is called a procedure, and every measurement here is called parameter. In IMPC, we have around 167 procedures and more than 3,800 parameters. For connecting the concept of reproducibility to the IMPC, we should first define this term. Here, here I have listed two definitions of the term reproducibility. First, the ability to be reproduced or copied. This definition is very mathematical and doesn't allow any degree of freedom. So we stick today to the next definition, which is the extent to which consistent results are obtained when an experiment is repeated. Okay, let's break the IMPC begin to end pipeline into the steps. The first step is the animal production. This step has some errors like measurement errors, data input errors, experimental errors, or overall human errors, etc. Data integration is the next step. As I explained in the previous slide, all data from 21 collaborators should be centralized and normalized into one single data set. Here, the source of error could be like dates formats, like European dates, American dates, and measurement unit errors like the American units or the British units or European units. And again, human error, such as uploading the wrong file or wrong format for the file. The next step is the data analysis step. As you see here, we already have the two error terms, E1 and E2, from the previous steps. And of course, we analyze the data statistically. So we allow another term, E3, in today's model. The last step is data presentation, that we display the data to the end users or allow exports and queries to the databases. As you see, in all of these steps, the E term is getting fatter and fatter. Now, if I want to model reproducibility in a statistics, I should say that a system is reproducible when all subsystems are to some extent reproducible. In other words, the sum of the error terms must be less than a maximum tolerance T. And for every step, we keep the errors below a threshold, as you see in the next line. Like here, E1 is less than a tolerance threshold of T1, and for E4 is less than a threshold 4. The beauty of this inequality is that we can add up something from the one term to the other one, or subtract something, some values from the terms. One important factor that we should consider in the IMPC pipeline is the scale of the data and feasibility of the statistical analysis in time. I will explain it in the next slide. Here, the chart shows data points produced by the IMPC from the first release in 2012 till the last release, data release 15 this year. The y-axis in this chart shows the total data points in millions. As you see in the current IMPC data release, we have 61 million plus data points, which is a little bit less than 67 million, the population of the UK. Imagine all of these data should be analyzed by the IMPC statistical pipeline as quick as possible. Then being on time and sticking to the schedules is an important factor that we should always keep in mind in the IMPC. Here in this talk, I'll focus only on two elements of the IMPC pipeline: the mass production and the data analysis, precisely reproducibility in the animal production and the measurements, and reproducibility in the statistical analysis, starting by the statistical analysis. All right, let's start by definition. When the input data is the same, statistical pipeline is reproducible when the same steps are taken to replicate the analysis. We know this in the IMPC, and to facilitate the reproducibility of the statistical results, we have two open source softwares, precisely R packages, to run the analysis. OpenStats, the new pipeline, and PhenStat, the old pipeline that we retired in 2020 last year. Before going to the comparison of the two pipelines, let's take a look at the methods that we apply to the IMPC data in this chart. We generally apply linear mixed model to the continuous data, Fisher exact test to the categorical data, and a heuristic method called reference range plus to apply to the continuous data when the linear mixed model fails or is not proper to be applied to the data. OpenStat and PhenStat implement the same models in their hearts, and I should say that OpenStat and PhenStat results are fully reproducible. Let's move to some improvements that we have done into the IMPC pipeline. As you see in this table, if we compare the statistical the computational time of these two softwares, we see a huge difference of around 20 months. Precisely, if we run OpenStats on 2.5 million plus data sets of the IMPC on an imaginary one core single core machine, it takes 2 months. However, if we apply the same to the same data, nearly the same data, PhenStat, the old pipeline, it takes 22 months. Obviously, all of these values are imaginary values because we haven't waited for 20 months for the results. So all of these are simulated on cluster computing, and it has a very difficult but very beautiful type of analysis for it. Anyways, then we have compared the result from two pipelines, and we have more than 99% agreement between the two results. This shows that we are still very reproducible in our pipeline. So we are back compatible and we are very reproducible. Most of our works will be published in or are published in +1 journal. So you can access to the journal and see the papers, or if you're interested in the OpenStats package, you can download it from Bioconductor. All right, summarizing the statistical analysis pipeline of the IMPC. We apply simple analysis to the data. This lowers the chance of creating irreproducible results, and it requires less computational resources, which is something that is of interest to us. And it leads to feasibility in time for the statistical pipeline. We have done very efficient coding and in-memory coding for the new OpenStats pipeline for the IMPC. This again requires less computational resources. However, because it's very complex, it increases chance of creating irreproducible results simply because it has more moving parts in it. But the good news is that it still contributes to the feasibility. We have done very good documentation, and everything that we produce is open source, as available from GitHub or any repository. Again, it contributes to lower the chance of creating irreproducible results, because more people could contribute to the source code and debugging of the pipeline. Let's talk about one more aspect of the IMPC pipeline. The IMPC team is a combination of biologists, statisticians, computer scientists, and web developers. When I finish my part, I pass the results from the statistical pipeline to the different groups to do the rest of the job, such as data display on the IMPC web portal, or putting data in the databases for the access points. This handling of the data could be a source of error. For example, if I pass the data with a non-friendly format to a developer, that would add more moving parts to their side and increases the chance of making mistakes or errors, and consequently put our reproducibility in jeopardy. Let's decompose E4 in the data presentation step into two errors: E4 handling plus E5. E5 is something unknown, we don't know it, the rest of the errors. In IMPC, we came up with a great solution to reduce this E4 handling error. This solution is called stat packet, and we have kind of shown that in our publication for OpenStats by putting a JSON object here. A stat packet, however, is not a JSON object, but JSON is the best representation of an stat packet. In the next slide, I will be explaining a stat packet in more details. What is a stat packet? A stat packet is a set of rules that facilitates the communication between data scientists and the other fields. Stat packet has some key properties. It should be human readable, it should be machine friendly, it should be compressible, and importantly, the statistical results in the stat packet must be fully reproducible by the data in the stat packet. As a result, passing the stat packet to the clients would be sufficiently enough to reproduce the statistical results in the IMPC. Just mentioning that, we have been working on the concept of a stat packet for years, and it is deployed to the IMPC portal. So pretty much everything related to statistical results in the IMPC web portal comes directly or indirectly from the stat packets. Let's move to another aspect of the IMPC pipeline, that is, IMPC data grows over time. The IMPC centers keep producing the animals, and we have frequent releases every year. As a result, we should always analyze the same data as the previous data release, plus some increments from the current release. Consequently, the statistical results in the IMPC should be reproducible within the current release and between the releases. And this is a big challenge for us. Let's take a look at the IMPC data to see what we mean by producing more data in every IMPC data release. Here we have an example of the experiments in the IMPC spanning from something, where's my mouse? Oh, okay, it's here. Something from 2013 to something around 2020. There are some facts in this data. First, all controls, orange dots and triangles, are clone of each other. This is a very important point in the IMPC. From the description of the experiment on the left, there are 2,287 animals included in this chart. 16 mutants, 8 males plus 8 females, and 2,271 wild types. This shows that we have great power for specifying the baseline population in the data. The other facts could be like the mutants are all collected on one day, March 6, 2014. And from that time, we only contributed to this experiment by adding more and more controls. In other words, in the upcoming IMPC data release, we will be analyzing the same data, the same mutants, plus a little bit of controls on the right-hand side. Or we can say that we have enough power for the controls, so we get a little bit of more precision for specifying the baseline. And of course, adding more data to any experiment means more resources required for the computations. And this is our first enemy. Okay, let's see anything else in this slide. Okay, no. Okay, but this is not the end of the story. In many instances, the control doesn't behave as expected. Looking at these two charts that are extracted from our recent publication in Bioinformatics, we see a kind of trend in our controls. Again, I stress that controls in the IMPC are clone of each other, so we shouldn't expect any variation, any trend in the data, but we always expect some natural variation in the controls. On the left-hand side here, you see a very downward trend in the controls, and in the right-hand side chart, you see a kind of a sinusoid, a seasonal effect in the data. We have some justifications for these behaviors. One could be the smell of the experimenter. If the smell of the experimenter changes, like changing perfume or looking after neighbor's cat or anything that has some, you know, could change some normal smell of the experimenter, it could have some effects in the animals, and it could produce some of these patterns. The other one could be the time of a year. Here you see some of these. Where's my mouse? Okay, here you see some of these variations happen around the Christmas time in January, February, and it could be a source of fluctuation in these dates as well. But anyways, we don't have any good justification for all of these behaviors. The scenario doesn't finish here. We have another scenario, which is not changing the trend in the data, but changing in variation. We call it change in variation because of unknown error, because spotting the source of variation here is really difficult. But there has been something in common in all of these experiments. The experiments span for a long period of time. From these two charts, from these two charts and more charts that we have, more behaviors or different behaviors that we observe from the IMPC data, we can make a conclusion that adding more controls, adding more wild type animals doesn't necessarily improve the statistical results. Let's take a look at some solutions that we have in the that we can propose for the IMPC. First, incorporating a seasonal effect into the statistical model. Second, assuming a complex structure for the random effect in the linear mix model. These two solutions look good because we treat the data with better model, but they add to the computational cost of the statistical pipeline, and doesn't actually help us because we have to reanalyze the data in every future IMPC data release. But there is a better option. How about we freeze the data over time? This is where the idea of soft windowing gets born. What is soft windowing? Soft windowing is a weighting method that gives more weights to the center of the window and less weights when we get farther from the center of the window. Here I quote a saying from Waldo Tobler: everything is related to everything else, but near things are more related than distant things. We have used these kind of idea in our soft windowing. The left-hand side chart shows the concept of windowing on one of our example IMPC datasets. As you see, the windows here, there are windows, but they are merged, and you see one single window here, are centered to the dates when the the mutants are measured these days that are shown with dashed vertical lines. And window expand to a point that, that an unknown variation starts. The right-hand side chart shows a very concrete example of soft windowing that and is extracted from our publication, Soft Windowing Application to Improve Analysis of High-Throughput Phenotypic Data. We published that in 2020 in Bioinformatics. If we assume the center of the window in the center of the blue lines, soft windowing helps to model the section of the interest in the data. However, if we apply a linear model to this data entirely, we see a dash black line, which is not modeling the part that we are interested in. I should stress that soft windowing is a method to specify weights only. That means all data are always included in the analysis. So in these two examples, not only this section of data is used in the statistical analysis, but the entire data is used in the statistical analysis. So the soft windowing differs from the hard windowing by the fact that we always use all the data. However, in the hard windowing, there are some boundaries drawn for the data, and only those data in that boundary are used. Okay. Okay, let's go to the other slide. As I mentioned, soft windowing is not a method to analyze the data. It accompanies the analysis method in IMPC linear mixed model to better specifying the baseline. In the heart of soft windowing, there is a weight generating function that requires two parameters to be set, the bandwidth L and the shape parameter K. Having these two parameters, the method is capable of producing multiple windows and merging them when intersect. In the left-hand side chart, you see the work generating function for different K and L. The bottom left chart shows clearly the merge of two windows when they intersect. As you see that for different K values, the shape of the parameter changes for the fixed bandwidth. And this is the beauty of soft windowing that could produce a variety of the window, not only the traditional window that we have in our mind, which is basically two vertical lines. On the right-hand side, I show you a video how soft windowing determines the best parameters for L and K by grid searching this space. The first step is to find the optimal bandwidth. So the algorithm searches the space for finding the best bandwidth parameter L. Here the bandwidth is set. Now the algorithm searches for different shapes of the window K to optimize the shape parameter. And here is the final result. Here is the final window to this data. And you see that these two windows have a little bit of merge. Okay, what are the cons and pros of soft windowing? Soft windowing freezes data over time. Then we do not require to reanalyze the data sets when the window is well specified. Soft windowing is computationally expensive, but it removes several rounds of reanalyzing of the data with each IMPC data release. Soft windowing greatly contributes to the reproducibility between and within the IMPC data releases. But as I pointed out, soft windowing could be expensive in terms of computation. And one more fact about that is that soft windowing is quite sensitive to outliers. Being sensitive to outliers is not actually something bad, it's something good for IMPC because we want to catch those bad points in our data. but it may not be ideal for other applications. Okay, after 3 years of discussion and hours of meetings and several rounds of comparison, simulation, and data analysis, we finally have the soft windowing in production in the IMPC data release 15.1, as you see here in one of the banners on top of the IMPC website. All right, going to the conclusion. IMPC is a rich resource for research in genetics and bioinformatics. We have contributed to the reproducibility of the IMPC statistical results by having a well-tested, documented open source statistical software called OpenStats, developing the concept of a stat packet as a protocol to communicate between data scientists and downstream researchers, developing soft windowing to freeze the data over time. Here are some resources for you that you can download the software or papers. The first resource is IMPC web portal, that is mousephenotype.org. You can download OpenStats paper or soft windowing paper from these two links. And the R packages are here as well. You can just Google them or use these links to open the URL. Finally, I take this opportunity to acknowledge some of my colleagues who helped building up a robust, scalable, and reproducible IMPC statistical pipeline, and of course, the whole IMPC consortium. Here are some of our names and the contact details for any collaboration, question, thought, etc. Thank you very much.